everyone, my name is Danielle Giangrasso. I'm a neuroscientist, and this is Sofa Science. So thank you all for joining me today. I am really excited to talk about the brain. But first, as this is Sofa Science, let me introduce you to my sofa. I actually recently got it, and I will say that so far, so good. It's really great, it's comfortable, and it even comes with office space for my research associate, Professor Rhubarb, who is hard at work, as you could tell. But yeah, so I study neuroscience, which essentially is the study of the brain all the way from the very microscopic cellular and molecular level up to the behavioral level of how we think and feel and interact with the world. And in particular, I focus on the neuroscience of addiction and how drugs of abuse can change our brain and change our behavior. But before we get into any of that, I was wondering, have you ever driven home from work without thinking about it, as if you were on autopilot? What about when you walk into your bedroom? Do you immediately turn on the light without needing to see where the light switch is? So these are examples of automatic behaviors that we've done so many times that we no longer need to think about doing them. And our ability to automate behavior is really important because by making routine behaviors automatic, it frees up so many cognitive resources that can be used for other tasks. Because, could you imagine having to put the same amount of thought and effort into driving as you did when you were first learning how to drive? It'd be a lot. It wouldn't be the worst thing for some of those drivers out there, if you know what I mean. But yeah, so we have this incredible and very powerful system in our brains that is guiding our behavior throughout our day-to-day lives. But this system can also get hijacked by drugs of abuse, and is one of the reasons why addiction is so difficult to treat. Because once someone becomes addicted to a drug, he will seek and take it as automatically as you and I turn on that light switch. But all of this first starts with drug abuse. And so people will abuse drugs for a variety of different reasons. They may take a drug because it makes them feel good, or they may take it because it relieves stress or reduces any emotional or physical pain, or maybe it just helps them fit in. But across all of these different reasons, there is one thing in common, and it's that taking the drug results in a positive outcome, whether it's giving them a good feeling or helping to reduce a bad feeling. And this is really reinforcing and is one of the reasons why people want to take a drug again and again and again. And as someone continues to take a drug over time, the brain will actually make the same shortcut as it does when we're learning how to drive. It will make these drug-taking behaviors automatic. And this is really important because once someone has reached the point of addiction, the element of choice is no longer there. They will take a drug automatically, regardless of the outcome, regardless of whether or not they still get a positive outcome, or even if they get a negative outcome, if it's harmful for their health or their family, and even if they want to stop. And so this is one of the reasons why addiction has spiraled out of control and has become one of the world's largest epidemics. And so neuroscientists have studied addiction from several different angles. They've looked at the genetic predispositions towards addiction. They've looked at the influence that our environment has and how the brain and behavior change. But what I'm particularly interested in is, how does the brain change as someone transitions from drug abuse to addiction? And so, interestingly enough, addiction has been studied in rodents for the past several decades. And this is because they will self-administer drugs in a similar way as we do. And so in these experiments, a rat is trained to press a lever in order to get a drug reward. And so we call this the drug-taking lever. And so this experiment has been done for every single kind of drug of abuse, and it's given us a lot of insight into addiction and how it works. But it wasn't until 2001 that Dr. Mary Olmstead and colleagues took a step back and realized that the human experience of addiction wasn't limited to just taking a drug, but it also included seeking it out first. And they hypothesized that this drug-seeking behavior could become just as automatic as the drug-taking behavior. And the way in which they tested this hypothesis was really clever. they introduced a second lever, a drug-seeking lever. And so here's how that experiment works. So first, a rat is trained to press a lever in order to get a drug reward. And so in this case, let's say cocaine. So after the rat has learned this, we then make it a little more complex. We introduce a second lever, the drug-seeking lever. And so here the rat will learn that when he presses the seeking lever, it gives him access to the taking lever. And then when he presses the taking lever, it gives him cocaine. And so the rat will do this for several weeks. He'll seek and take and seek and take and practice this behavior a lot over time, the same way as we do when we're first learning how to drive. We then ask, has this cocaine-seeking behavior become automatic? And the first step in answering this question is to actually take away the cocaine itself. And so here the rat sees the taking lever, but this time, no matter how many times he presses it, he finds that he can no longer get cocaine. And so the rat will do this for several weeks until he's learned that this lever no longer has any value for him. It no longer gives him the cocaine. And then we see how this affects his cocaine-seeking behavior. So here the rat is given access to the seeking lever, and we record the number of times that he presses it. And so this data helps us ask the question of, now that the rat has learned that he can no longer take cocaine, will he continue to seek it or stop seeking it? And these two different stages actually reflect whether or not the rat is abusing cocaine or if he's addicted to it. And so in the case of drug abuse, again, an individual will seek a drug in order to get a positive outcome. So in our experiment, in a rat who is abusing cocaine, if we take away this cocaine outcome, he will actually stop pressing the seeking lever because there's no cocaine. Again, this cocaine-seeking behavior was done in order to get the outcome. So if we take away the outcome, they're going to stop performing that behavior. Okay, so now in the case of addiction, again, individuals will seek a drug automatically regardless of the outcome. So in our experiment, rats who are addicted to cocaine, when the cocaine outcome is taken away, they will actually continue to press the seeking lever because the seeking behavior has become automatic. And it's no longer sensitive to whether or not they actually get cocaine. And so these studies have actually shown us that approximately 60% of the rats abuse cocaine and 40% of the rats are addicted to it. And so now that we're able to look closer at these addictive behaviors, the next question is, what's changing in the brain? And there are a lot of different things that are changing in the brain, but I think a really interesting place to start is within the part of the brain that makes behaviors automatic. And this region is called the striatum. And so within the striatum, our ability to automate behaviors is mediated by the neurotransmitter glutamate. So you may have heard of neurotransmitters before. They are the chemical signals that our brain cells or neurons release in order to communicate with one another. And you may have also heard of the neurotransmitters dopamine and serotonin. They get all the hype all the time. But glutamate is a really important neurotransmitter, and it's actually critical for learning and memory. And so here's an example of glutamate signaling. Here we have a signaling neuron that is releasing glutamate onto a second receiving neuron. And if you look closely at this point of communication between the two neurons, this is called the synapse. And so when the first neuron releases glutamate into the synapse, it will bind to the second receiving neuron and generate a signal. And then this signal is sent to the next neuron and the next and the next. But this can't go on forever. So over time, glutamate gradually gets cleared out of the synapse and recycled back up into those neurons so it can be released again and again. And studies have actually shown that within the striatum, when a behavior becomes automatic, glutamate signaling is stronger. And so one of the ways in which glutamate can become stronger, glutamate signaling can become stronger, is by having glutamate remain in the synapse for a longer period of time. And this is because glutamate now has more time to bind to that receiving neuron and generate more signals, which makes glutamate signaling stronger over time. And so what I want you to remember here is that the longer glutamate is in the synapse, the stronger glutamate signaling becomes. Longer, stronger. Got it? Okay. So what do we know so far? We know that addiction can be classified as having automatic drug-seeking behavior. And we know that within the striatum, When a behavior has become automatic, it's associated with stronger glutamate signaling. And we know that one way to make glutamate signaling stronger is to keep glutamate in the synapse for a longer period of time. And so taking all of this together, this leads us to our hypothesis, where rats who are addicted to cocaine, again, rats who have this automatic cocaine-seeking behavior, glutamate will stay in the synapses of the striatum for a longer period of time as compared to rats who are just abusing cocaine. Okay, so we have this great hypothesis and we're excited, we're ready to go, but now how do we measure glutamate signaling? How can we possibly look into the brain and measure this very tiny and very fast neurochemical process? And one of the ways actually comes from jellyfish. And so there's a certain species of jellyfish that has what is called green fluorescent protein. And this protein makes the jellyfish glow green. And scientists have actually been able to take this green fluorescent protein and modify it in a way so that whenever glutamate binds to it, it generates a fluorescent signal. And so in this way, it acts as a glutamate sensor. And so what we're actually able to do is then take this glutamate sensor and put it into the brain and onto the actual brain cells themselves, and then use this sensor to measure glutamate signaling in real time. So in our example, let's say we put this glutamate sensor into the striatum. We then can measure glutamate signaling in the striatum by taking this piece of this brain tissue and putting it under what is called an epifluorescence microscope. And so this is a microscope that detects and records fluorescence. So we put our striatum underneath this microscope, and then we use what is called a stimulating electrode to stimulate the neurons of the striatum, cause them to release glutamate to the synapse, and then once glutamate is in the synapse, it will bind onto our glutamate sensor and generate a fluorescent signal. And so here's an example of the glutamate sensor at work. And so here we're in the striatum, and we have our stimulating electrode, and we stimulate those neurons, it releases glutamate, and we get this big, beautiful increase in fluorescence. And so if that didn't impress you enough already, what we can actually do is take this signal and analyze it in order to understand more about the kinetics of glutamate signaling. And so here's a graph of the fluorescent signal you just watched. We're looking at the change in fluorescence intensity over time. And you can see that at first there's a very low level of fluorescence. There's not a lot of glutamate signaling, so it's very low. We call this the baseline. But then when we stimulate the neurons and they release glutamate, it generates this big increase in fluorescence that gradually goes down and returns to baseline over time. And this part of the graph we're really interested in. It's called the decay of fluorescence, or how long it takes for that fluorescent signal to go back down to baseline. And this is because it reflects how long glutamate stays in the synapse, which corresponds to the strength of glutamate signaling. Okay, so now taking all of this together, what do we expect to find? So in our rats who are abusing cocaine, again, their cocaine-seeking behavior hasn't become automatic yet. So when we look into the striatum and look into this part of the brain that automates behavior, we don't expect glutamate signaling to be any different because there's no behavior that has become automatic. So we expect it to look like this. However, in rats who are addicted to cocaine, again, their cocaine-seeking behavior has become automatic. And we know that within the striatum, when a behavior has become automatic, glutamate signaling has become stronger. So we expect that. And we know that one way to make glutamate signaling stronger is to have it stay in the synapse for a longer period of time. And so what that would look like in our results is that glutamate, the fluorescent signal would take a longer amount of time to decay, a longer amount of time to return back to that baseline. And so this is what we expect to find, but the great thing about science is that no matter what we find, no matter whether or not our results support our hypothesis or they go against our hypothesis, it all brings us one step closer to the truth and one step closer towards understanding addiction. Because if we can identify what's changing in the brain, we can then target those changes with new medications. And once we've developed those medications, we can combine them with cognitive and behavioral therapies for addiction in order to treat this disease in a more comprehensive manner. And this is one of the things that I really love about neuroscience, how these changes in neurochemistry of genes and proteins can all translate up and change our behavior. I think it's incredible how everything is so connected. And this also applies to science in general. It's not just a subject, it's a way of seeing the world, a way of seeing this interconnectedness, of seeing cause and effect, and testing our observations. And it's just a great way to experience the world around us. And so I used a lot of resources to make this talk, which I've included if you're interested in checking out. Also, if you're interested in learning more about addiction, I highly recommend checking out the National Institute on Drug Abuse's website. And of course, if you or someone you know is struggling with addiction, you can contact the Substance Abuse and Mental Health Hotline, because although addiction is very complex, there is help out there for you. And if you're interested in learning more about science in general, you can check out the Natural History Museum of Utah, either online or in person. And Utah is just an incredible treasure trove for archaeology, paleontology, biology, zoology, and this museum does a really great job of taking all of these local finds and making them into exhibits so that we can all learn about the world around us. And so also be sure to tune in next week for Professor Rhubarb's talk on the thermodynamics of hairballs. Does a hairball in motion stay in motion?